Hello everyone, welcome to my YouTube channel Raj Botany. If you have not subscribed my channel, please do subscribe it. Friends, today we will discuss on a very important aspect of plant pathology, that is fungal diseases. One of the very important fungal disease of the wheat crop, that is rust of wheat. So let's see. Mainly, the rust of wheat is caused due to the pathogen called as Puccinia graminis. In figure, we can see here it's a symptom. Reddish brownish spots are developed on the stem as well as the leaf portion. These are the oblong pustules on the both abaxial as well as adaxial side of the leaf, on the stem, and on the glume portion of the leaf. The fungal mycelium produces thousands of spores that rupture the leaf epidermis and emerge out as a powdery rust-colored piles of the uridinospores. A particular spores are produced in this disease. That spores are termed as uridinospores. These uridinospores has the capacity to cause new infection and damage same plant as well as different plant. Stem rust spores are much darker in color than the leaf rust spores. Here in figure we can see the spores appeared on the stems are much darker than the spores appeared on the leaf. As the plant matures, the pustules produces the black spores known as the teleospores. The black colored spores produced by the Puccinia graminis, their spores are now termed as the teleospores. And due to its dark black tilia, the dark black color of these teleospores, this disease is termed as black rust of wheat. Let's see the pathogen biology. This Puccinia graminis pathogen is the obligate parasite. That means for its survival and its reproduction, it required the living host tissue. Means it cannot be a saprophyte. Saprophytes means the organism which grow on dead and decaying organic matter. But as far as this Puccinia pathogen is concerned, it required living host only. In the absence of the living host tissue, this Puccinia survive in the form of teleospores, and that teleospore adapted itself to survive if there is the absence of living host also. The Puccinia graminis is heteroecious. That means it required two unrelated host plants such as wheat and barberry to complete their life cycle. Puccinia graminis is heteroecious. Next term is macrocyclic. Puccinia graminis is macrocyclic, means it produces all the 5 types of spores: basidiospores, pycniospores, esiospores, uridinospores, and teleospores. This figure we can see these are the teleospores. These are the basidiospores. These are the pycniospores, and these are the uridinospores. In the diagram, in this figure, we can also see in this picture the tilia and uridia on the wheat stem and leaves. When you take the transverse section of the wheat stem, it shows the appearance of this tilium. The uridinium produced on the wheat as well as stem. From the uridinium, there is a development of the teleospores, and these teleospores, it is actually the two-celled, and from this two-celled teleospores, there is a development of the basidiospores, and this is a basidium condition. And from the basidiospore, there is a production of this pycnia, and inside this pycnia, there is a development of the pycniospores. And from this pycnia, these are actually produced on the upper side of the barberry leaf, and exactly opposite to this, what you can say, the pycnia. Here we can see here these are the pycnia, this is the section of the barberry leaf. These are the Puccinia, and on the opposite side of this the Puccinia, there is a development of the Asia. Asia is a plural, singular is Asium. Here this is a figure of the Asium. This is the lower side of is a barberry leaf. Here we can see the cluster of Asia on the lower surface of the barberry leaf. These are the more focused portion of the lower surface of the barberry leaf, and this is a microscopic view of this single Asium, and these are the Asiospores. From this Asiospores again, uh, from the ejection from the barberry, the Asiospores again infect to the fresh wheat plant, and this disease cycle goes on. The complete life cycle of the Puccinia graminis involve the barberry as well as the wheat plant. The stem rust infection on wheat plants convert the urodinospores to the teliospores. Teliospores remain attached to the infected plants and are commonly left in the field in the form of straw residue of the crop. These teliospores are very specialized structure for the survival of this pathogen, and it remains dormant in the field until the upcoming spring season. These teliospores of Puccinia graminis have two haploid nuclei. One is plus mating type, another is minus mating type, and during the dormant period, these nuclei fuse to produce the single diploid nucleus. These are the plus mating type and these are the minus mating type. Then next, the diploid nucleus divide. The plus and minus mating type nucleus fuse to produce the diploid nucleus. That diploid nucleus divide by meiosis to form the 4 haploid nuclei. And during the spring season and in the warm wet weather, this teliospore germinate, and when they germinate, it produces another type of spores. That spores are named as the basidiospores. From the diploid nucleus, after meiosis happening, there is a development of the basidiospores. Each of the 4 haploid nuclei per teliospore. These are the teliospores. This figure we can see the teliospore, and this teliospore actually produces the basidiospores. That's why it is written here. Each 4 haploid nuclei per teliospore cell migrate to one of the 4 developing basidiospore. In this figure we can easily see, yeah, this is figure. This is a teleum, and from teleum, after the meiosis, there is a development of 4 basidiospores. The mature basidiospores are forcibly ejected and carried away by air current. If they reach a susceptible barberry plant, especially the leaf of the barberry plant, these basidiospores germinate and penetrate the leaf. Basidiospores produces the pycnia. Pycniospores are produced in a sugary nectar within the pycnia and function as the male gamete. These are the pycnia, and inside the pycnia, the production of the pycniospores. They consist of a little more than a nucleus to fertilize the receptive hyphae of another pycnium. These are here we can see the presence of the 2 pycnium. This is the mating hyphae of one pycnium, and this is the mating hyphae of another pycnium, and these 2 hyphae are fused to each other, and these 2 hyphae are compatible hyphae. The receptive hyphae function as the female gamete. One hypha function as male gamete, another hypha function as a female gamete. The sugary nectar released by pycnia helps spread the pycniospores. This pycnia produces the sugary nectar, and that sugary nectar helps to spread the particular pycniospores. Exact role of this sugary nectar that attract the insect, and that insect often visit to the several pycnia in a succession and fertilizing them. Self-fertilization is totally absent in the Puccinia graminis because only plus mating type pycnospores can fuse with minus mating type of receptive hyphae and vice versa. When pycnium has been fertilized by pycnospores, its cells quickly change from their original haploid uninucleate state to a new dikaryotic condition. Initially, before their fusion, there is a presence of monokaryotic condition, but after their fusion, there is a appearance of the dikaryotic condition. The fertilized structure becomes an acium. Now it is called as a acium, which produces chains of aciospores. Here, this is the lower surface of this leaf, and from the lower surface of the leaf, there is a presence of this acea, singular acium, and inside this acium, there is a development of the aciospores. This is a cup-like acium. Each aciospores contains two nuclei. Now, each aciospores bear two nuclei and infect to the. Now, this is ready to infect the new wheat plant, not barberry plant. On barberry plant, only there is a presence of this pathogen is only up to the development of the acium. After the formation of this acium and aciospores, this aciospores later on ejected and infect to the new host, that is wheat, not barberry. Pycnia typically form on the upper side of the barberry leaves, and acea form within 5 to 7 days after fertilization on the lower side of the leaf directly below each fertilizer pycnium. This acium are produced exactly below the pycnia. This is a previous figure. These are the pycnia, and exactly opposite the pycnia actually present on the upper surface, and exactly opposite to this pycnia, development of the acea or acium. This is overall disease cycle of rust of wheat. When basilospores infect to the host, the production of the pycnium, and this pycnia exactly opposite to its pycnia, there is a production of this acium. In acium cup-like structure, acium is a cup-like structure. It produces the aciospores, and these aciospores present actually on the barberry leaf. Acium are actually present on the lower surface of the barberry leaf in the cluster form. And after the development of the aciospores, these aciospores infect to the new wheat plant, and through the stomata, this aciospore produces the germ tube and infect to the wheat plant. There is a production of the spores on the very lower, exactly low to the epidermal surface. From the spores, there is a development of the two-celled teleospores, and these teleospores, after their germination, they produce the basidium, and on the basidium there is a production of the basidiospores. These basidiospores now infect to the barberry leaf, and this disease cycle is going on. Means for the completion of the disease cycle of Puccinia pathogen, it's required the two different hosts, barberry and wheat plant. Let's see the disease control. First, buy disease-free seed and healthy plant material, so far as to avoid this Puccinia pathogen. Then plant disease-resistant varieties when available. We have to plant disease-resistant variety if we have to avoid that pathogen onto the wheat plant. Choose a plant that fit the site. Keep plants healthy. Always keep the plant into the healthy condition. Destroying previous wheat plants and volunteer wheat by TDS or herbicide, because if there is a residue remain into the field condition, and during the next crop season, if there is a pathogen of the previous crop, that pathogen certainly infect the newly seeded crop. That's why that is recommended that is a complete destruction of the previous wheat plant. Then adequate balanced fertilization based on the soil test should reduce the possibility of the severe yield loss due to the rust. High nitrogen rate without sufficient phosphorus and potassium may increase the rust severity, reducing the grain yields. Dear subscriber, thank you for your patience hearing. If you not subscribed my channel Raj Botany, please do subscribe. Please share this video. Thank you one and all.